So today we are going to start the new topic, that is the plant alkaloids. So as the name suggests that these agents are isolated from the plant sources, right? And hence the name is the plant alkaloids. Yeah, if we have to simplify these plant alkaloids, these agents they differ in their structures and the mechanism of action if we compare these plant alkaloids with the previous classes of the anti-cancer agents. But these agents they share multi-drug resistance mechanism, right? They share the multi-drug resistance mechanism. Means what? That is, these drugs given with the other classes of anti-cancer agents to treat the cancer. These plant alkaloids are the cell cycle specific agents and they are classified into the four different classes. That is Vinca alkaloids. The examples are Vinblastine, Vincristrine. Podophytotoxins, examples are etoposide and the teniposide. Camptothecins, examples are topotecan and the irinotecan. And the taxanes, examples are Paclitaxel and the Docetaxel. So these agents today we are going to cover in the plant alkaloids. So the first class of the plant alkaloids is the mitosis inhibitors. So the mitosis process, it is depend on structural and the functional viability of the microtubules. The polymeric heterodimers consisting of isotypes of the alpha and beta tubulin proteins. So these microtubules, it is made up of alpha and beta tubulin proteins. These distinct but nearly identical 50 kilodalton proteins lie adjacent to one another within the tubule and they roll up to form the open pipe-like cylinder akin to a hollow peppermint candy stick. So these alpha and beta tubulin, they placed like a cylinder, means it look like a cylinder because of their adjustment or because of their places. They look like a pipe-like cylinder akin to a hollow peppermint candy stick. And the gamma tubulin, it is present at the center of the microtubule. So the alpha and beta tubulin lies like a well. It is lies like a cylinder, and at the center of that cylinder, gamma tubulin protein is present. Now during cell division, because this is what mitosis process. So during mitosis process, cell divides. So during cell division, this tubulin undergoes intense sporadic and alternating periods of structural growth and erosion known as dynamic instability. The proteins alternatively polymerize and depolymerize through guanosine triphosphate and calcium dependent process. So who is going to polymerize and depolymerize? That it requires, of course, it requires some enzyme, that is the guanosine triphosphate, and also calcium is required for the polymerization and the depolymerization process. The polymerization involves the addition of tubulin dimers to either end of the tubule, although the faster growing end is more commonly involved. Polymerization results in tubular elongation, whereas depolymerization results in the shortening of the structure. The frantic alteration in structure facilitated by microtubule associated proteins ultimately allows formation of the mitotic spindle. So whatever the polymerization and the depolymerization process ultimately results in the formation of the mitotic spindle and the attachment to chromosomes that are prerequisite to the cell division. Later on, this mitotic spindle it attached to the chromosome, which is prerequisite to the cell division. The inhibiting the essential hyperdynamic changes in microtubular structure results in mitotic arrest and the apoptosis. So the two general chemical classes of the mitosis inhibitors have historically been marketed for the treatment of cancer. So these are the Vinca alkaloids and the Taxanes. So the first uh, drugs of the mitosis inhibitors are the Vinca alkaloids. The examples are Vinblastine and the Vincristrine. The Vinca alkaloids, they are extracted from the leaves of Catharanthus roseus, also it is called as the periwinkle, and it is originally investigated for the hypoglycemic properties, but later on it is found that it possesses some anti-neoplastic activity also. Structurally related compounds derived from Vinca rosea, that is the vinblastine and the vincristine. Vinorelbine is a semi-synthetic derivative. Despite their structural similarity, there are significant differences between them in regard to clinical usefulness and the toxicity. So in short, these Vinca alkaloids, they are isolated from the leaves of the Catharanthus roseus, but initially it is investigated for the hypoglycemic activity, but later on it is found to show antineoplastic activity also. Whereas there is the third drug called as a vinorelbine. It is a semi-synthetic derivative, means it is not isolated from the leaves. So it is a semi-synthetic derivative, vinorelbine. Regarding the mechanism of action, as we know that these Vinca alkaloids, they bind to the tubulin and inhibit the polymerization process, and which disrupt the microtubule structure. Okay, so this is the mechanism of the Vinca alkaloids. Now, if you check out, this is the structure of the Vinca alkaloids, right? This is the structure of the Vinca alkaloids. So it's a very big structure of the Vinca alkaloids. So we just summarize the structure. Okay. So the Vinca alkaloids, they are the complex structures. Vinca alkaloids, these are the complex structure, and it is composed of two segments. The one is the catharanthine portion, or it is also known as the valbanamine portion, and the another one is the vindolin portion. Okay, other one is the windowing portion, and both the portions are connected by a bridge. Okay, so the portion which is present at the top, it is called as the catharanthine portion, and the portion which is present at the bottom, it is called as the windowing portion, and both the portions are essential for high affinity tubulin binding. So the three commercially available anti-cancer alkaloids, they differ. Now, which are the three? Vinblastine, vincristine, and the vinrelbine. Okay, so these three commercially available anti-cancer alkaloids, they differ in the length of the alkyl chain, bridging position 6 dash and the 9 dash position. Okay, so all the three alkaloids, vincristine, vinblastine, and the vinrelbine, they differ at these alkyl position, right, which is present in between 6 dash and the 9 dash of the catharanthine moiety. So either it is a methylene or it is ethylene portion. Now in this case, it's a ethylene portion because there are two carbons. But in few alkalides, that is either in vinblastine or vincristine or vinrelbine, it is either methylene, or in some cases it is ethylene. Also, all these three alkaloids, they differ in the substituents at position 4 dash. Either it is olefinic linkage or it is tertiary alcohol. Also, they differ at the N1 windolin nitrogen. So either the substitutions are methyl or the substitutions are the formyl. So this is how all these three Vinca alkaloids, they differ with each other. So where they differ, that is they differ at position 6 dash and 9 dash, means this chain either it is methylene or the ethylene. Then they differ at the substituents at position 4 dash, either it is olefin linkage or it is tertiary alcohol. Also, they differ at the N1 of the windolin nitrogen. So either it is methyl. Now in this case, check it out. It is methyl. In this case, it is formyl. Okay. Although subtle, these structural changes lead to significant differences in chemical spectrum, potency, and the toxicity of the particular Vinca alkaloids. It is known that the acetylation of either hydroxyl group, okay, so there are several hydroxy group in which is present in this Vinca alkaloids. So if we acetylate this hydroxy group, then it destroys the antineoplastic activity, and the reduction of the vindoline olefinic linkage, it attenuates the antineoplastic action. Then C18-methoxycarbonyl. So where it is C18-methoxy? So over here, C18-methoxycarbonyl, right? Methoxycarbonyl. So C18-methoxycarbonyl, and the stereochemistry at position 18- and 2- are also believed to be critical to activity. Okay. So the C18-methoxycarbonyl and the stereochemistry at position 18- and 2- are also believed to be critical to the activity. So if we differ at this position, so instead of the C18-methoxycarbonyl, if we insert C18-ethoxycarbonyl, or if we change the stereochemistry at the 18- and 2-, then there will be a loss in the activity, or the activity may reduced or decreased. Then the active site of these Vinca alkaloids is close to the residue 339 and residue 390. So where these residues are present? So these residues are present on the microtubules, the residue 339 and residue 390. So the active site of the Vinca alkaloids, it is close to the residue 339 and residue 390 on alpha and beta tubulin respectively. So once these Vinca alkaloids once bind to these your alpha and beta tubulin, then it does not allow the elongation of the microtubule chain. The Vincristrine bind most tightly, whereas Vinblastine has the lowest affinity. Okay, so if we compare the binding of the Vincristrine and Vinblastine, so Vincristrine bind most tightly as compared to the Vinblastine, because Vinca alkaloids, they enter cells by simple passive diffusion. Unbound Vinblastine, because Vinblastine bound the binding of Vinblastine is less as compared to the Vincristrine. So whatever the unbound Vinblastine, it may more extensively taken up into the tissues. Vincristrine, however, is cleared more slowly from the system and has the longest terminal half-life. Yes, because Vincristrine, it is bind tightly to this tubulin. So it is excreted very slowly, or it is cleared very slowly, and hence it's having the longest duration of action. The Vinca alkaloids, they undergo O4 deacetylation to yield metabolites. Okay, it undergo O4 deacetylation to yield metabolites equal to or more active than the parent drug. So where is the, yeah? So this one. So there is a acetyl group, O acetyl group. So if we remove these acetyl groups, so means what, which reaction takes place? That is the deacetylation reaction takes place. So that is the metabolite of the Vinca alkaloids, and is having the similar activity to the parent drug. Now this vinblastine is used in the treatment of various cancers including Hodgkin's disease, lymphocytic lymphoma, histocytic lymphoma, advanced mycosis, fungites, advanced testicular carcinoma, and Kaposi sarcoma. It is also used in treating choriocarcinoma and breast cancer when other therapies have failed. The vinblastine is part of the ABVD, that is adriamycin, bleomycin, vinblastine, and the dacarbazine. So vinblastine, it is a part of this ABVD regimen and is used in the treatments of the Hodgkin's lymphoma. So this is what all about the your Vinca alkaloids. Now the next one is the Taxanes. Okay, next one is the Taxanes, as these all are the plant alkaloids. In the plant alkaloids, first we covered the Vinca alkaloids. Now the next one is the Taxanes. Now the Taxanes, initially isolated from the bark of the Pacific yew called as the Taxus brevifolia. But now it is produced by the semi-synthetic route to a natural precursor called as 10-D-acetyl dacatin 3, which is found in the leaves of European yew, Taxus baccata, which is a renewable resource. Now these taxanes are cell cycle specific. That is, they act on G2 phase and M phase of the cell cycle. Regarding the mechanism of action, these taxanes bind reversibly to the beta tubulin subunit, which promote polymerization process, and it stabilizes the polymer rather than its disassembly. Thus, they shift the depolymerization polymerization process to the accumulation of the microtubules. The overlay stable microtubules formed, they are the non-functional, and chromosome desegregation does not occur. This result in the death of the cell. So over here it is given the mechanization of the taxanes is given by the diagrammatic view. So these our taxanes, they bind to the beta subunit of the tubulin, and thereby they promote the polymerization process. And if they promote the polymerization process, then the microtubule chain get elongated. The microtubule chain which is formed, which is useless, so the cell will die. So the taxanes, they bind to the tubulin at a site distinct from the Vinca alkaloids. The first site was located at the N terminus and involved residues 1 to 31. The second site involved residues 217 to 231 of the tubulin. And although these two sites are widely separated in the primary structure, they are close to each other in the tertiary structure. So these are the different sites of the beta tubulin, or where the taxanes are bind. So which are the sites? That is these taxanes. The first site of the taxanes, it is located at the N terminus, and it involves residues from 1 to 31. Whereas the second site, it involves the residues from 270 to 231 of the beta tubulin. Okay. So the binding site is located on the luminal side of the microtubule and located in the middle of the tubulin subunit. The Docetaxel bind to the same site with greater affinity. Binding of taxanes at low concentration results in stabilization of the microtubule and further prevent the depolymerization process. So what actually that is the mechanism of action of the taxanes? That is, they prevent the depolymerization process. But at higher concentration, polymerization is enhanced and the normal equilibrium between free tubulin and the polymer is modified. The taxanes inhibit both treadmilling and the dynamic instability, and cells are most affected in the M phase when the microtubule dynamics are undergoing the greatest change. Mitosis is blocked at the metaphase anaphase boundary, and cells undergo apoptosis. So this is the structure of the taxanes. At the left side, this is the structure of the Paclitaxel, and on the right side, the structure of the Docetaxel. So chemically, they are the diterpenoid taxanes. It consists of 15 membered. So check it out. There are total 15 carbons in the ring of the taxanes. So it is a 15-member tricyclic taxane ring system, which is fused to an D ring called as the oxetane ring, and it contains an esterified beta-phenyl isoserine side chain at C13. Okay, so this is the beta-phenyl isoserine side chain which is present at the C13 of this tricyclic taxane ring system. Now these taxane antineoplastic, they differ in substitution pattern at C13. So if you check the structure of the Paclitaxel and the Docetaxel, so there is only variation at the C13 substitution, else otherwise the structure is same. Okay, so they differ in substitution pattern at C13. So either it is benzamido. So check it out over here in the Paclitaxel, it is a benzamido ring, or it is a tertiary butoxycarboxamido, tertiary butoxycarboxamido ring system. Also, they differ at the C10 position. Either it is a secondary alcohol, or it is a acetate ester, or it is a methoxy ether. And also they differ at the C7 position. Either it is a secondary alcohol or the methoxy ether. So all these taxanes, they differ at these different positions. So they differ at the C13 position, at C10 position, and at C7 position. The taxane ring system is often conceptualized as having northern and the southern halves, right? So they are having by hypothetically, it is considered that they are having the two halves. One is the northern half, and the another one is the southern half. The southern segment, it is critical to the receptor binding. So the southern segment of this paclitaxel and docetaxel, it does not particularly bind to the receptor. Whereas the northern section, it ensures the proper conformation of essential functional group. So the northern section, so which are the different functional group which bind to the receptor? So they include the C13, so this one, the C13 isoserine side chain with its C1 carbonyl, free C2OH and C3 benzamido or tertiary butoxycarboxamido groups, right? So what are these C13 position or the C13 groups? So these C13 side chains, they are responsible for the binding to the receptor, right? Also the benzoyl and the acetyl esters at the C2 and C4. So Oriers at C2 or a benzoyl ester and and the C4, so over here this one acetyl ester. So these C2 and C4 groups, they are also responsible for binding to the receptor. The paclitaxel interact at the beta tubulin binding site in a folded conformation. So this paclitaxel drug, the paclitaxel drug, it bind to a beta tubulin binding site in a folded manner, that is either in a T shape or in a butterfly shape, which places the C2 benzoyl and the C3 benzamido groups in close proximity. So this C2 benzoyl and this C3 benzamido, this group come close with each other when they bind to the beta tubulin subunit, because they bind in a folded manner. So once they bind in a folded manner, so this group come in close proximity, this C2 group and this C3 group. Their independent intermolecular engagement with a critical beta tubulin histidine residue perfectly positioned between them keeps them from interacting with one another. Whereas the oxetine ring, so this is the oxetine ring of this paclitaxel and the docetaxel, which is a D ring. The oxetine ring of the taxanes, although capable of enhancing receptor affinity through hydrogen bonding, is believed to serve a more critical role in properly orienting the C4 acetyl moiety interaction with its hydrophobic binding pocket. So this oxetine ring system is also responsible for the binding to the receptor which is present on the beta tubulin. So this taxane ring bind to the receptor of the beta tubulin by forming a hydrogen bond. Then the C1 hydroxy, so this one, C1 hydroxy. So the C1 hydroxy also promote conformational stability through intramolecular interaction with the carbonyl oxygen of the C2 benzoyl moiety, right? So this C1 hydroxy group, it also promote conformational stability through intramolecular interaction with the carbonyl oxygen of the C2 benzoyl moiety. So it form interaction with this oxygen, because in short these drugs bind to the receptor in a folded manner. So all the groups which are far away from each other, they come in the close proximity with each other. So proper orientation, they bind to the receptor and then it shows the action. So this is how they mention. The areas of the paclitaxel structure where steric influences are most critical to receptor binding have been identified. So this is all about the metabolism of the paclitaxel and the docetaxel. So this paclitaxel is hydrolyzed into the 2-hydroxy paclitaxel. So where is the metabolism? 2-hydroxy paclitaxel. So over here, 2-hydroxy. So over here the hydroxylation reaction takes place to form a 2-hydroxy metabolite. So it is a 2-hydroxy paclitaxel. Then also paclitaxel is metabolized into the 3-hydroxy paclitaxel. 3-hydroxy. So over here, now this is a 3' position, and over here the hydroxylation reaction takes place to form a 3-hydroxy paclitaxel, right? Then also paclitaxel is metabolized into the 6-alpha hydroxy paclitaxel. 6-alpha hydroxy. So this is a 6th position. So over here the 6-alpha hydroxy paclitaxel, which is the metabolite of this drug. Then another metabolite is a 3-6-alpha dihydroxy paclitaxel. So 3 So this is the structure of the paclitaxel. So this is the our 13 the side chain which is present at the 13th carbon. So this is the 3-1, 2, 3. There are 3 carbons in a structure 1, 2, 3. So this is a 3-hydroxy paclitaxel, and this one is the 6-alpha hydroxy paclitaxel. So collectively, this is the metabolite which is form of the paclitaxel. That is a 3-6-alpha dihydroxy paclitaxel. So there are various metabolites of the paclitaxel. Whereas the docetaxel is metabolized into a single metabolites. Whatever the docetaxel is having the tertiary butyl ring. So over here, the hydroxylation reaction takes place to form a hydroxy docetaxel.